Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
MacConkey Agar explained. Table of Contents. Introduction. Composition. Principle. Preparation. Result interpretation. Uses. Limitations. Introduction to MacConkey Agar. MacConkey Agar was the first solid differential medium to be formulated. It was developed in the 20th century by Alfred Theodore MacConkey. MacConkey Agar is considered the earliest selective and differential medium designed for the cultivation of coliform organisms. The medium is specifically used for the isolation and differentiation of non-fastidious gram-negative rods. It is particularly useful for identifying members of the family Enterobacteriaceae. It is also applied in the study and differentiation of the genus Pseudomonas. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Composition of MacConkey Agar. Peptone, pancreatic digestive gelatin, 17 grams. Proteose peptone, meat and casein, trigram. Lactose monohydrate, 10 gram. Bile salts, 1.5 gram. Sodium chloride, 5 grams. Neutral red, 0.03 gram. Crystal violet, 0.001 gram. Agar. Treton, femgram. Principle of MacConkey Agar. Pancreatic digest of gelatin and peptones, meat and casein, supply essential nutrients, vitamins, and nitrogenous compounds necessary for microbial growth. Lactose monohydrate serves as the fermentable carbohydrate source. Crystal violet and bile salts provide the selective property of the medium by inhibiting most gram-positive bacteria. Sodium chloride maintains the osmotic balance of the medium. Lactose fermenting strains produce red or pink colonies. The red coloration results from acid production during lactose fermentation. This acid leads to absorption of neutral red and a subsequent color change of the dye when the medium's pH drops below 6.8. Non-lactose fermenting bacteria such as Salmonella, Proteus, Pseudomonas aeruginosa, and Shigella are unable to utilize lactose. Instead, they metabolize peptone, leading to the production of ammonia. This raises the pH of the medium and results in the formation of white or colorless colonies. These colonies often appear golden to brown with dark centers, and the surrounding agar remains relatively transparent. Modification of the medium is possible by replacing lactose with other sugars to study fermentation patterns of gram-negative bacteria. Example: replacing lactose with sorbitol helps in differentiating enteropathogenic Escherichia coli serotypes, such as E. coli O157:H7. In this case, non-sorbitol fermenting E. coli produce white circular colonies on MacConkey Agar. Preparation of MacConkey Agar. Suspend 49.53 grams of dehydrated MacConkey Agar medium in 1,000 milliliters of distilled water. Heat the mixture until it reaches boiling to completely dissolve the medium. Sterilize the medium by autoclaving at 15 pounds pressure, 121 degrees Celsius, for 15 minutes. Allow the sterilized medium to cool to a temperature of 45 degrees Celsius to 50 degrees Celsius. Mix thoroughly before pouring into sterile Petri plates. Result interpretation on MacConkey Agar. Escherichia coli, pink to rose-red colonies, often surrounded by a zone of precipitated bile. Enterobacter and Klebsiella, mucoid, pink colonies. Proteus, colorless colonies, frequently showing swarming growth. Salmonella and Shigella, colorless colonies, sometimes with the medium appearing orange to amber. Pseudomonas, irregular colonies that range from colorless to pink. Uses of MacConkey Agar. Used for the isolation of gram-negative enteric bacteria. Helps in the differentiation of lactose fermenting from non-lactose fermenting gram-negative bacteria. Applied in the microbiological examination of foodstuffs. Suitable for direct plating, inoculation of water samples to perform coliform counts. Employed in the examination of milk and dairy products. British Pharmacopoeia recommends this medium for the subculture and identification of Escherichia coli. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Limitations of MacConkey Agar. The colonial characteristics observed on MacConkey Agar provide only presumptive identification of organisms. Subculture and confirmatory tests, such as biochemical tests, are required for final identification. Certain bacterial strains may grow poorly, or may even fail to grow on this medium. Incubation under increased CO2 conditions can reduce the growth and recovery of several gram-negative bacilli. Some strains of Proteus may exhibit swarming growth on this medium, which can interfere with interpretation. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!